Have you ever heard of jumping genes? Well, they are actually called transposons. And one of the most common ways we can see them is in the variation in color of leaves and flowers. They also help control the color and shape of fruits.
There's a lot more to them. Transposons are snippets of plant DNA that can copy themselves into new regions of a genome. There are two types of transposons, class 1 and class 2. Today we will be talking about class 1 transposons, also called retrotransposons. Retrotransposons are copied into an RNA intermediate during transcription, which is then copied back into DNA by reverse transcription using a reverse transcriptase that is encoded by the retrotransposon. The DNA is then inserted back into the genome. It is important to note that the original retrotransposon does not leave the DNA. Transposable elements make up a large part of the genome, but have a tendency to become silent to counteract any unwanted behaviors they might have on a plant. Some of these behaviors include deletions and harmful mutations in the genetic code. Some of the silencing mechanisms involved are hypermethylation of cytosine residues and by the formation of self-reinforcing silencing loops via siRNA. However, the activation of transposable elements can be activated via environmental stresses. One example of this is the family of Reiter retrotransposon found in tomato plants. In a recent news article, a study from SLCU was cited which described the functionality and uses of Reiter. This retrotransposon, which was once considered junk DNA, is now believed to have a role in drought stress resilience in tomatoes. By experimentally activating Reiter, the researchers were able to quantify the amount of Reiter RNA intermediate to determine drought response. After analysis of Reiter activation, it was also shown that DNA methylation had been drastically reduced and the presence of siRNAs was non-existent. The activation of these Reiter retrotransposons leads to possibilities in genetic and epigenetic variation.
So why would genetic variation in plants matter to humans? Well, as we mentioned before, Reiter is activated via drought response. By using transposon-directed mutagenesis, Reiter can be used in the creation of drought-resistant crops. Even beyond environmental response signaling, other retrotransposons found in the tomato family, like Messi, have been shown to respond to developmental signaling, opening up more areas of research into crop mutations. These types of mutated crops are necessary for the future of agriculture as climate change continues to affect crop yield and thus requires more resilient variants. This may sound like something far into the future, but current research has demonstrated success in engineering heat-tolerant Arabidopsis. A study involved in working with the onsen retrotransposon demonstrated a clear connection with retrotransposons and heat stress. Onsen is capable of bypassing silencing by incorporating itself into heat-responsive elements recognized by the heat stress defense factors. The host plant is unable to respond to stress factors without losing control over onsen. This results in the retrotransposon's transcription and activation when subjected to heat stress. Its activation can be beneficial to the plant by rendering additional plant genes as stress responsive, generating overall genetic diversity. But Arabidopsis and tomatoes are not the only plants that have been found with transposons. Other economically important crops, such as quinoa, beetroot, and rapeseed, also contain transposons similar to Reiter. This points to the possibility of improvements for multiple crop species, as well as other types of mutations in addition to drought and heat resilience that could be possible. Now it is time for our conceptual question. We have talked about how transposons can be very helpful, but what are some ways transposons could negatively impact plants, the environment, or humans?